Good evening. As every year, we devote the lecture in the beginning of the year in memory of Daniel Amit. And I'm happy to see Dalia here, and I'm happy to see some people from the physics department here. And I would like to say a few words before Shaul gives the lecture. And um, I can say this because I'm old enough, but I'm on the limit of being able to say this because I'm young enough. And that is because I met Danny when I was at the end of my postdoctoral studies, exactly at the end of my postdoctoral studies. And then I learned what you will see now, that Danny was among the initiators of ICNC. ICNC is the Interdisciplinary Center for Neural Computation, which is the beginning of what we are today, which was the reason why we are today, which is the spirit of what we are today. And it was really a great experience. I say this in a complete sincerity. It was a great experience as a PhD student to see what is happening. I was a PhD student towards my end of my work. Then I went for my postdoctoral studies. When they started here, I came back. They already had the ideas. They, I mean, Hanoch, Dani, Moshe, I forget anyone. Chaim came you came at 1986, 2-3 years later. It is after I came back from post, which was 282. Okay, so they already had the ideas. They brought Chaim into the team, and it started. It started a collaboration between theorists and experimentalists. And as you all know, all those who sit here, this is what we are today. Few words about Danny, the things that I best recall of Danny, and the things I always felt about Danny in any interactions that we had. So as I said, the dramatic move was from doing mechanical statistics or something like that, moving to the real world, to interdisciplinary neurosciences. This was in 1980 or so. Dalia, I hope I'm right. He is among the founders of modern neural networks, and this was the experience that I got as a student. Suddenly, people talk in models term about what we do. It was a big honor to be invited to talk to the people in the physics department about our experiments, and they didn't just listen and said, okay, go, you are dealing with nonsense. They listened. And then it was difficult to create the understanding of what they mean. It took time, but it was a great success. So this was the beginning. Then he was a thinker, an open-minded person, and that's why he took such a central role in establishing these collaborations between experimentalists and theorists, mainly from the physics at the time, then later on from computer science and other disciplines. He was advocating all his life, since 1980 at least, to the important role of theory in neuroscience, and the results came. Like the book that he published, you can see even today is being sold, modeling the brain functions, the world of neural attractors. He was not only so passionate in his way of thinking about science, but he was passionate about everything he was doing. So he had a role, for example, in expressing himself in a very vivid way about anything, any event in the political world, in the society, in the interactions between the society in Israel and the world, to science. He was the example of a passionate, idealistic in all respects. So we honor him, we'll remember him for as long as we are here, and hope that the next generation, will continue to remember. And for now, I want to invite Shaul, who is the best possible lecturer in Dani Amit lecture, because The most significant collaboration, I think, of Daniel Amit with the experimentalists in Jerusalem was the collaborations that was established between Danny and Shaul group at the time. Some of them are here, for example, sitting here, Udi Zohari, and this is how it all started. And Volodya is also sitting here, and he certainly remember the work that he did with Danny. So Shaul, thank you for agreeing to give this lecture.
Thank you very much. Yeah. I'm supposed to go now to my own. Yes. I am very happy and honored and moved to give the Daniel Amit Memorial Lecture. It was actually 7 years ago that I was at with Volodya at the Society for Neuroscience, and we were about to present a poster of work we had done together with Danny when we heard the sad news. These were our presentations, both with Danny. I immediately printed out a picture of Danny, put a black border around it, and the dates. And everybody who came to the poster to speak about science and the findings and so on reflected on Danny and what he was and the important work he had done. Danny was a great man, deeply rational in his thoughts, and a man of true integrity. He was a great and brilliant scientist, leading, as Elon said, the physicists to neural network revolution, writing with Hanoch and Chaim the most influential papers in the field, authoring the textbook, interacting with Moshe Abeles, Tali Tishby, and many others here to unite researchers in the various fields to form ICNC and ultimately ELSC. His integrity was not easy or simple. He was willing to pay the price for his political beliefs to the end. He was quite unique in being a non-lawyer, active in the Association for Civil Rights in Israel, which actually my wife Annette headed for a while, also a non-lawyer, and found it very difficult. He was sent to military prison when he refused to serve in a war that he believed was illegal and humanly wrong. And he was willing to let his activism encroach on his scientific work, not on his science, but on his scientific work. And there are stories which you will hear about from others about his receiving and not receiving scientific prizes due to his political views. I was happy and honored to work with him in the last years of his life. He was an integral part of you went too far. A 7-year grant from the ISF, a center grant, more than $2 million, which he was central in. It included physiology, fMRI, psychophysics, computation, and this is just a small group of us with Danny in the middle in Eilat at the Israel Science Society for Neuroscience meeting. So today I want to talk to you about insights to the field of working memory, but they're Danny's insights, and unfortunately I'm a poor representative of his brilliant thinking, but I'll do my best to tell you about it. One of the things that Danny said was so important was simulation, and that simulation lies somewhere between theory and experiment. What did he mean by that? That theory is based, first of all, on the prior experiments, certainly for its function and for the parameters that one puts into it. Theory determines simulation algorithms. Simulation is easier than wet experimentation. That anyone who's done physiology knows, and certainly monkey physiology. It's easier to do simulations than it is, but it takes more pre-thought. It's harder to do the right things. In wet experimentation, you say, oh, we'll just look, we'll find something. Results of simulation aid in finessing the theory, and the finesse theory aids in designing further experiments that critically support or reject the theory. And I hope you'll see this in my talk today about how this interaction between theory and experiment works and how it worked in the work that I did together with Danny. I'm sure you're all aware of this, as I say, because you've been educated or brainwashed at ICNC. The theory that Danny was attracted to was attractor networks, which are very rich in two important aspects. First of all, long-term silent memory is a synaptic structure which is set up by repeated image presentation. Okay? We see an image, some neurons respond to it, and they interact by Hebbian learning, whatever it is, and build a new synaptic structure. And that synaptic structure is silent. And that's what the memory is of what was in the world. Second is working memory. The reverberate, if you get these neurons working, then the reverberation, which is the network, the attractor, is initiated when a threshold number of network neurons is activated and maintained by excitatory intra-network connections. And this allows for all kinds of new aspects, like recognition of a partially occluded image. Okay, you still know it's me, even though half my face is covered. And images often appear together or sequentially might share neurons, and this thus initiate recall of associations. And one attractor might facilitate initiation of another, reflecting ease of QRST and difficulty of TSRQ. How many of you can say the alphabet backwards? Right? Sorry, this pen doesn't work. No, help me.
We can all do it, but very slowly. You do it faster than we can.
Right? Not good?
Now in English.
Now, why can I do this? It's very simple, because I say Tikanta Shabbat Vatiza Kabanatea Tivita Perisha. In other words, if you've got a mnemonic, then you can do it. But if you don't, you can't. Okay. Good. Just wanted to test you. Okay. So all this is in, okay. So we will confront attractor networks with delay match to sample task. And we're going to ask 3 questions and slowly answer them. First, where does the theory lend an explanatory basis for the experimental phenomena? That's what theory, that's what we want theory. 2, where does the theory allow for the phenomena if one allows for further ad hoc assumptions, even without experimental proof that this is the theory that's correct? And number 3, where does the theory fail or need serious adaptation to be consistent with experimental phenomena? Okay, so we're starting with the experiment. We go to theory. And then we ask, well, where do we go from there? Good. Okay, delay match to sample task. What we do in delay match to sample task is we present a cue, call it A, then we'll test. If it's again A, then it's a match. That's going to happen 50% of the time. Maybe it's going to be something different, a non-match 50% of the time. If the response to the match, then it's a hit. If there's no response, it's a miss, false alarm, correct rejection. And the brain must somehow remember the cue in order to compare it with the test image. And this is called working memory. So here's an example. There's a sample, a delay, a match, or a sample, a delay, a non-match. And the question is, will the subject, the participant, the monkey or the person, be able to do this correctly? And how is this recalled from here to there? Okay. The intriguing finding was that in a number of experiments, a number of researchers found delay activity. Delay activity meaning there was a high level of activity of the neurons between the cue and the match. So this could work as a memory. In other words, this response started with the sample, continued, maybe decayed, maybe other things happened, and eventually And this could be a memory which was somehow compared with what happens here. Okay, 3 papers by Yasujiro Miyashita, which came out in the 80s, in which he did 3 things. First, he found that when he did exactly this delay match to sample task, these were his pictures, he found, as I said, delay activity. Here's the first cue, delay test, and the cue turns on the response, and it continues to respond until the test, and then sort of goes off in this case. And if it's a different neuron or a different stimulus, then there's very little response. Okay? Delay match to sample. Number 2, he looked for long-term memory of paired associates. What do I mean by paired associates? He trained them that if this was going to be the cue, this was the match that they should respond to. If this was the cue, this was the match. And the idea was that these were randomly chosen forms. Okay? There's no association between them. And what happened after long training? All these experiments require the monkey to be trained for months until they're able to do this properly. And here's one neuron, probably his best neuron that he ever found, where when this was the stimulus, this was the response during the stimulus, this was the continued response during the delay, and here was during the presentation of the match. The same neuron, when presented with the pair, did not respond, but during the delay period slowly got more of a response, and then when the first one was presented, responded. Okay? Here's the second best neuron that he found. It responded to this one, and a little bit of delay, and nothing when the pair. And this one, when you present the same neuron, the pair, in fact, all 4 of these are the same neuron. This is the best stimulus, and this is the second best stimulus. When you present the pair, it was even inhibited, but slowly some response, and then a response there. So the delay activity responds not only to the stimulus that was put there and remains in memory, but to what is brought up to memory because the monkey has learned to associate with it. Number 3. Number 3 has to do with what happens when associating different things. So here the stimuli are these pictures. And what happened was, in this case, for some reason, Miyashita decided to train the monkeys on delay match to sample using 100 different pictures. Okay? And then, when he was finished, and he always used the same 100. Every day the monkey came in, picture number 1, and then either a match or non-match. Picture number 2, match or non-match. Picture number 3. Okay? And when he was finished, he started recording. And what he found was, strangely, the gray are the response to these 100 neurons, these 100 stimuli, and one neuron responding to all of these, best to this one, but also to the one next to it and the one next to it. Now, these were chosen randomly in a random order, so there should not, have been any association between them. And yet, somehow the neurons learned what was coming next, and the same neuron responded to sequentially appearing pictures. Okay? So 3 things. Delay activity between cue and test, activity predicting pair associates, neurons learn to respond to sequentially presented images. So now, what the question that we're going to ask is, how come they learned these associations and where? But before I go on with that, I want to say something about delay activity and the strange thing that happens in science. Delay activity was the cue that maybe attracted neural networks with something really happening in physiology. But now that we say that maybe attracted neural networks are happening in physiology, we no longer need the delay activity. And recently, Shaul Druckmann, who received the Daniel Amit Prize a few years ago, showed that neural circuits underlying persistent representations can do that even if they don't have delay activity. And I want to show you something of his work. Here. Play. Let's see if this works. Okay. So he's saying that you don't need persistent activity in one neuron in order to get this response. So if you have two images, the standard understanding was that you have delay activity. Okay? But it's true that Miyashita found a few cells with them, and we found a few cells with them, and another 10 people in the world found a few cells with them. But most cells don't have strong delay activity. Okay? And even the ones I showed you, the best ones, were a large response there and then quite a small response here. Okay? Okay. And so Shaul says, well, if we found all kinds of things like that, sometimes the activity goes up, and sometimes it stays, and sometimes it's low, and sometimes it goes down. So what does that mean? Can complex time-variant neural activity underlie persistent stimulus representation? And the answer is yes, but only under certain conditions. Usually, if you have two neurons representing one point in the world, then this one, I'm not going to be very good at this, so bear with me, can represent that. But if you change one, that this one goes up and this one goes down, then it represents something different. So you can have two neurons. Together, they can represent one thing, but if they go to... Okay. But what happens in the real world is that from the thalamus to the cortex, and also in other places, there's a tremendous expansion in the number of neurons. So we're overdetermined. And once we are overdetermined, we have a lot more play. So that this point in image, underdetermined, this point in space can be represented either by a lot of activity in number one, or the sum of these three activities. Okay? So all you have to do is set up the network so that it'll do this. Okay? And so he does that. And he does it in the following way. He makes sure that when this one goes up, the other two goes down, the other two go up, and vice versa. And then the same point is always represented by Daya, thank you. While if it's not set up that way, then this point just keeps moving in space. So only under certain conditions can you have that the same representation will happen there. And he then goes on to say what are those conditions. And the conditions are that while this has what we want ultimately is represented by different parts of the world in certain ways, all the neurons that it feeds into represent parts of that in different ways. And then all you need is that when this goes up, that goes down, and vice versa. And you can read his papers that showed all this, and I'd like to go on if I can. Okay? So what we need ultimately is that the change will be such that it'll be 0 because the one goes to the other. Okay? So basically, the way A1 feeds into A2 is such that you get the sum of A2 and A3 will be A1. And therefore, if A1 goes down but these go up, then you are left with the same thing. Okay? So the reason I'm bringing this here is to say that even though we talk about delay activity, it's not necessarily delay activity in one neuron, but rather the group of neurons together have to somehow represent the same thing. Okay, that's that. We can go back to myself and go to R2. Okay. Here we go. Okay. Okay. So Donnie asked, why should it be that these same neurons represent a different number of images just because they happen to be presented in sequence during the work that I run? And the answer came from work which Ehud Zahari did. Udi did this together with Volodya and Stefano and Elisha Berman. And what they found was the following. let me go straight to the result. And the result is this. That if you look at one trial and then the next trial, and we go from sample to test, and there's delay activity between them, maybe there's also delay activity from the test that goes on to the next sample. Now, in 50% of the cases, the test will be a match, and therefore will be the same as the sample. So in 50% of the cases, what we're doing, otherwise it's going to be a variety of things. In 50% of the cases, there'll be delay activity which will connect the first sample with the next sample. Just like there is from the sample to the test to be able to compare them and to see if it's a match or not match. So that same neuron will continue that way. And Udi found that indeed, that here's an example, a cell which has strong delay activity between the stimulus and the match, also has activity which continues between the match and the next test. This is the period before the next test. Okay, the next test will come right after that. And here's another one, and another one. So what Udi found was that these attractors are able to make associations between not only the sample and the test, but also between the test and the next test coming after. And the result is that IT neurons seem to remember not the last stimulus seen always, no matter if it's important or not important. Here it's important to remember it between the stimulus and the test, but from here to here it's not important. Okay? And yet we remember. So maybe we're always remembering and comparing things that are happening. Looking at what Miyashita had done with viewing the same sequence of stimuli in one particular sequence, Donnie said, well, why don't we try that and see how we get a result? And Tanya Orlov said, okay, but she wants to do something more interesting than that. And so she did the following test. First, she presented 3 images, first A, then B, then C. And then she put the 4 images on the screen, the 3 images on the screen, together with a distractor. And she asked the monkey to point to A, B, C, and then the distractor, and not the distractor. Okay? So here's the task. 1 stimulus, 2nd stimulus, 3rd stimulus. Touch them in the right order on the touch screen. Okay? Let's see if you can do it. Okay, here are the stimuli. 1, 2, 3. Okay, what's the order? No? Oh, it came up. Okay, 1, 2, 3. There we are. Good. All right. So you weren't fast enough. Okay, good. But in order to satisfy Dani's desire for a fixed sequence, she said, okay, she'll take 30 stimuli, and she'll present them always in the same order. A1, A2, A3, and then the 3 together with a distractor. B1, B2, C3 with a distractor, and so on. Okay, always in the same order. And we'll see if there is built up some sort of associations between the different stimuli, maybe between B3 and C1. Okay. The distractor was taken randomly from the others. So, for example, it could be D1, D2, D3, and then those 3 together, let's say, with G2. Okay. Okay. What happened was most of the errors, okay, here's a very important thing in psychophysics, you have to look at the errors, not only at the success. Most of the errors happened when the monkey touched not the right ones in the right order, but touched the distractor when the distractor was from the same category, 1st, 2nd, or 3rd, as the one he was supposed to touch. So let's say this was 2nd, then he would touch 1, 2, 3 instead of 1, 2, 3. That means somehow he was learning something about the stimuli. So Tanya said, well, let's see what we can do about that. If he is learning that, what happens if instead of showing him C1, C2, C3, and then the 3 with the distractor, let's just put up C1, C2, C3, and the distractor. What will he touch? If he's learned that this image is always a 1st, and this one is always 2nd, and this one is always 3rd, we don't have to show them. He's not using working memory. He's using long-term memory. He's learning an association between the images and their category. So she did that. And they got it mostly right. So she said, okay, let's try another one. What happens if we shuffle? We'll do C1, F2, D3. And put those up with a distractor. So that was pretty good also. Except, of course, the distractor was from one of the categories, and there they didn't know which was which. There was no way they could know. And then what she did was say, well, let's do the both of them together. We won't show them the samples, and we'll just put up 4 things from, okay? And then they completely didn't know the difference between the one which was the distractor and the correct one. When they were doing D1, D2, D3, and the distractor without showing them, they had learned that these items are associated with each other as well, not only with their category. And therefore, D1, D2, D3 was better than D1, F2, D3. Okay. So, sorry, sometimes this jumps. If the test was A1, A2, A3, and C1, then first of all, as they were learning these stimuli, and they start out of chance, of course, they learn, but they learn to do the right one, but they make a lot of mistakes. They've here got to 60-40 for a long time, about 500 trials where they're trying to do this. And then they got it, that A1, A2, A3 are associated not with C1. However, if it's C2 rather than C1, then for the first touch, they'll always do A1 very quickly. They'll learn. Okay? So they are learning the correct category faster than they're learning the associations among them. Okay? Okay. So what's the order?
1, 2, 3.
Which is one?
1, 2, 3.
You're remembering the place, but I moved them around. One, thank you. 1, 2, 3, right? Okay. Just as a test. Okay. 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 So, Tanya used a variety of tests to find out the strategies used by the monkeys. A fixed sequence of images, all the strategies are available. Without the samples, they can't use working memory, but they can use the triplet associations. Shuffle the images amongst the triplets. They can't use the triplet associations, but they can use working memory. Shuffle plus no samples, no strategy available, and they get it 50-50. And so, when the basic task, they get it correct, very high, but they're still making 20% errors touching a distractor when it's of the B1, right? If there are no samples, they're still doing pretty well, but it's still reduced, even though they have the association between D1, D2, D3, so D1 should belong. Shuffled, F1, A2, C3, and B1. Well, they can use working memory because they were shown the samples, and they still get it. So this is about halfway between this and chance. No samples and shuffled, there's no way of knowing if it was F1 or B1, and therefore it's 50-50. Okay. On the other hand, if it's we're talking about the distractor, but they're supposed to be touching the third. Let's say this was the third touch, then they all get it right. They have no problem once it's the wrong category. Okay. So they learn all 3 possible stimulation characteristics: online identity and order of presentations, ordinal number categories, associations between sequentially presented stimuli. So the Daniel Amit dogma says all associations are learned only after establishing delay attractor network activity. It's when they meet, when the attractor activity meets the next sample, that's how you get the associations. And the maintained reverberation allows for interaction between different stimuli and their multiple aspects. Okay. Let's move to the next set of stimuli. And this is work by Volodya. And what he said was, okay, we've got this delay match to sample, and we know that there's delay activity which can underlie it. But what happens if we do something a little bit more tricky? Because in that case, what happens if we add more stimuli, we can have cue, non-match, 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 wait for the match. Okay? That's the first expansion of the delay match to sample task. And then the delay activity would have to wait all through this until the match comes up. But Bologna demanded even more of his monkeys. He said, we're not going to tell the monkey which is the sample. It could be the first one, but in this case, for example, it's the second one. This is the sample which is going to be matched. So the monkey has to remember all the stimuli all the time until finally there's one which matches what came before. Okay? And in that case, what that means is we're going to need multiple delay activity or multiple attractor activity until finally one of them matches the sample. So here's one example. There can be 5 stimuli, and the second one in this case matches. Now, there are 4 things that can happen. When the match comes up, if the monkey responds, it's a hit. If he responds when it is not yet the right one, it's a false positive. If he does not respond when he should, then it's a miss. And his non-responding in the meantime are correct rejections. Okay. So the first thing he did was take 16 fixed images, teach the monkey these 16 images, train them very well on them, and then do the experiment where there can be sample 1, sample 2, sample 3, sample 4, cue and match. In other words, this is where the match is. This can be any stimuli. Or the cue could be here and the match here, and these are other stimuli. So there's a difference between the distance. And as it turns out, the earlier it is, in other words, the more distance, the earlier it is, the worse the performance. So performance depends on how many images there were in between. They're disturbing. Okay? Here are the 16 images he used in one series of experiments. So you have the Q in this case is the second one, and D is 4 in between. And here are the set of results. And the results show 2 things. First of all, as we get closer to the end, we get higher performance. And number 2, the more there are in between, the worse it is. So 2 things are happening. Number 1, the greater the cue to test distance, the more images in between, the worse the performance. And second of all, the more there are in the trial, it can be, let's say, 3 back. 3 back would be from there, 1, 2, 3. From the next one, 1, 2, 3, and the next one, 1, 2, 3. So they're getting worse. Okay? So it depends how many that came before as well. Okay, and we know the current attractor network. I never even did this here. And, excuse me. And so if we do the learning properly, we'll get the synaptic metrics, which will associate these things. Now, Bologna said, well, let's try another experiment. He said, okay, the dogma says you can only learn, you can only be able to remember the cue till the match if you have built up an attractor. Building up an attractor takes something like 100 times of viewing that image. What if we take new images that the monkey has never seen before? So he went to the internet, took down 10,000 images, and said, okay, we're going to use these. In that case, the only time there's a repetition is when it's cue and match. If it's cue and non-match, no repetition. Okay? And again, he's doing a whole series, and they can match to any one of them. Okay? Sample. Okay? And we have all this. But now, there's not going to be delay activity. There's not going to be delay activity because this is the first time they've seen it. So they shouldn't be able to do it. Right? And of course, what happened was that not only could they do it, they did it even better. fewer mistakes, more hits. Somehow, they were able to detect, and if we compare the two, these are 16 well-known stimuli, and these are 12,000 novel stimuli. Okay? How could they do it? That's the question. And then we remembered that actually Standing had done an experiment in 1973 where he showed 10,000 pictures. He went to college students at Columbia, showed them 10,000 pictures. It took him a few days to show it to them. And then a month later, he came back and he showed them 2 pictures and said, which one was amongst the 10,000? What percentage did they get right? 90%. Interesting, huh? Okay, let's try it. Okay, where am I going? Okay, so here what we're going to do is the standard Bologna task. Okay, standard Bologna task. Are you ready? Okay, so what you have to do is scream loud and fast as soon as you see a repetition. Okay? Very good. Okay, let's try it again. Fewer get it. When it's the first one, it's harder. Because the first one can never be a match. I got you that time. That's called a catch trial. Okay. I have a few more. Please keep going. Very good.
No. No.
I think we have 2 more. It's okay. You didn't get that one. This one, okay. You've gotten tired. Okay. 2 more, and then we're ready for the second test.
Yeah.
As you know, for the monkeys, all these pictures are meaningless. You've never seen a Hanukkah or Queen Elizabeth. Okay. And now the standing test. I'm going to show you 2 pictures. One you've already seen and one not. Which one is the old one? You're supposed to answer right or left. Okay?
Right.
Right, we have seen.
Left. 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 Right. Left. Right. Right. Left. Right. 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 Right.
Left.
Left.
Right.
Left.
Now, aren't you amazed? Aren't you amazed? You've seen these pictures once for half a second. And you were not told to remember them. Standing actually told his subjects, you're looking at this, he gave them a few seconds, so you should really learn them. You weren't. You were told, look at them and remember them for a few seconds later when you're going to see a match or a non-match. And yet these complex things you got, and we're able to differentiate, and there's work done recently, mainly by Ortoliva at MIT, about how different does it have to be. You know, you saw glasses and so on. how different does that be to be able to differentiate and say which yes and which not? Okay. Okay. No attractor, right? Okay. So that's the question. Let me go on. And, we're getting there. So, how do we model familiarity memory? How do we model that we were able to say, I've seen this and I've never seen that? We walk down the street, we see somebody in the street, and we say, I know this person, and I don't know that one. And I don't remember who he is, and it's certainly at my age. You don't remember who he is and where you've met him and so on, but you know. How does that work? So Donnie suggested the following. He said, just like when you start out with a network of neurons, a whole bunch of neurons, which each one responds to a different stimulus. When you see that stimulus, then all those neurons build up synaptic interactions amongst them. They have more positive interaction between them and less positive interaction between them and those who are not responding at that time. And so you build up the attractor. And it takes about 100 times that you've seen it to build it strong enough that you get the reverberation and the delay activity. But, he said, what happens if just after once you get the same kind of Hebbian learning? But that single, what happened? You're not seeing any of this. I'm sorry. That's because I went the wrong way. Sorry. You didn't see it. Why didn't somebody tell me? Okay. But what happens if after one time, a single stimulation, there will be a tiny trace of the now familiar stimulus in the synaptic matrix. And maybe that tiny trace is enough to differentiate between something I've ever seen before and something I've never seen before. Okay, so that's the model. Simple network, reproduces phenomenology. Okay, I'm not going to go into it. And therefore, if we say there is no delay activity, but the response to this same stimulus, since I've seen it before, is a little bit higher than it is to the others. And my picture is simplistic because there has to be something that, neurons respond stronger and less, and there has to be something which will normalize it and so on. So we know that this is bigger than some normal and so on. Okay. Good. Okay, conclusion. Monkeys' performance with novel stimuli is surprisingly better than with very familiar stimuli. Monkeys should not have all the advantages of full attractor network for images seen only once. They can know, for example, if I had shown you two pictures and asked you which one came before the other one, you probably would not remember. Or did they come in the same trial or not in the same trial, etc. All those things you're not supposed to be able to remember according to the model. Okay, let me just put this in terms of other things that we do in our lab. And that is, we think there's a difference between rapid effects and gradual effects. Rapid effects pop out in search, which you probably all heard about. Conjunction search is slower. It takes more time. Rapid is learning. Affects transfer to new conditions, if it's easy learning. High-level learning. Effects are specific to training conditions in other ones. It's slow learning. Conscious perception is a rapid capture of the gist, while slow integration of the details takes longer time. And finally, now we're saying perceptual memory, one-shot familiarity memory, comes very, very quickly, while context associations might require more time. Okay. But that's only beginning the story, because we said that we can learn a lot about behavior from correct hits, but also from false alarms. So let's look at the false alarms. Okay. And let's look now at the false alarms of these two possibilities. Here we have 16 images learned by the monkeys. How many false alarms? Well, if the picture came, the same image was shown the preceding trial, one trial back, then about 10% errors. And then there's about 2-3% errors if it's further back. Okay. But what about novel images? Well, novel images, there shouldn't be any repetition. But we snuck some in. Okay? We did catch trials. We took some images from the preceding. 1 in 10 trials, there was an image that came from some preceding trial. In that case, he got 80%. He responded to the one that came from the preceding trial, and 60% from the 2 trials back, and so on down the line. So the monkey knows if it's familiar or not familiar, and that's what he's using in order to respond. Is it familiar? Is it not respond? So good performance is due to indiscriminate familiarity memory. Okay. So here's the comparison of the two. They're doing better on the novel images, but at the expense of not being able to reset between trials and to say, I don't want to respond if it happened in the preceding trial. On the other hand, without the reset, we can measure how long does memory take. And there is a decay of memory. In this case, under these conditions, so for 3 minutes, he can remember things. Further back, he's not making this error. Okay. Now we did another thing. Volodya said, well, what happens after he's been trained for novels? Now we'll do the fixed set. Will that make a difference? And actually, it doesn't make a difference. The fixed set after novels is pretty much the same as the fixed set first and the fixed set after trained. More or less the same. There are a little bit more errors for one back. We don't really know why. Now, what about novels after the fixed set? And here there's a dramatic change. This is just the same, but the errors went away. Here's a monkey that first was trained on the fixed set, and he established a reset mechanism. And somehow he's able to move that reset mechanism also here to the novels. Okay? So when we compare the two, now this is comparing novels after fixed and fixed after novels, and they're about the same. But here's the essential one, which is the novels first or the novels after the fixed set. And all this tremendous number has gone away. Maybe he's learned, well, I don't know how to put things in terms of monkey thinking. But monkey thinking might say, well, I've now learned from the fixed set that I'm not supposed to respond if it came from the preceding set. I had to learn it there because there are only 16 images, and a lot of them are repetitions, so I know I'm not supposed to do that. So I've been trained not to respond if it came to a previous, so I know how to do that here also. But what does that mean? How's he doing it? Okay. If we have a model that says, well, there's no delay activity, but there is some change in the synaptic structure, that little change, well, how does he get rid of that? Or does he not get rid of it? Okay. Training with a limited fixed set of images initiates a reset mechanism to avoid the large number of false positives, which would otherwise ensue. Performance with novel images does not require reset, and it is not developed, as seen with a large number of catch image false alarms. Reset mechanism initiated by fixed image testing is transferred to subsequent testing with novel images. I have one more thing to say to you before I finish. And that is, we're going to move now to see what happens with people. We're going to compare monkeys and people. So you've seen this. This is the monkey, the fixed set, and this is the monkey fixed set false positive. Here is the monkey's novels first, and here's all their false positives. This is the monkey after the fixed set performance. Okay. Now let's go to people. People can do better than monkeys. And therefore, we use longer trials. Okay? So the trials go up to about 10 images per trial. So they might be sample, 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 Q, sample, 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 match. Okay? So there's a lot more, and it's more confusing and so on. And they do pretty well, but not as well as the monkeys. The monkeys got it more than 90%. People get it about 80%. Okay. And what about errors? Well, false alarms, they get, something like 30% if it's one back and 20% they also got false alarm errors. Okay. Now, what about novels?
What age are the
Most people in psychometric and psychophysical experiments are college students. So in America, they're between 18 and 22. In Israel, they're between 30 and 15. Okay, so this is people's novels first. And here is people novels first performance, yes. So this is novel set for people when they have not had prior experience with a fixed set. And nevertheless, they have very few false positives. So it seems that people have a reset mechanism. They understood, perhaps. They somehow have a reset mechanism even before they have trained into it with a fixed set. Now we asked, and Volodya said, okay, but have they reset so that they've forgotten all those images? Or are they able to discriminate between images of the preceding trial and this? They said, okay, let's ask them now, after they've done this experiment, to now please tell us if they've ever seen these images before. So please respond not only when it is a match in the same trial, but also when it's a match from a preceding trial. And then they got 80%. So people are somehow able. What? It's positive. I know it's called false positive, but it's a correct positive. Okay, because we asked them to do it. We asked them, please make false positives. Okay. Yeah. Okay. So, what is the result? Monkeys have no inherent reset. No reset for novel images. They establish a reset when they're trained with a small fixed set. But once it's established, it transfers the reset to the novel image. People seem to have an inherent reset for both fixed sets and for novels. They recall the images despite reset and avoiding false positives. Give me 2 more minutes, I'm finished, and then I'll ask for questions, okay? So we need a new explanatory model. Okay? We need to somehow understand how this works, that people can remember and yet not respond. The basis for the model. I've already said that attractors allow for association. That one of the important things that we're able to do is associate and to connect different stimuli. And as Tanya showed, to connect them with the category in what order they were, or we can connect them with where they've seen, where they're positioned, etc. So attract activity recalls multiple images. The attract activity enables associations between sequential images, between image pairs, between images and their order categories or other such categories. So maybe the 2 new assumptions that we're going to have to make if we want to model how people do this task is that attractors may be set up even with one presentation. I said there's only a little bit and therefore it's not an attractor. Well, maybe it is, or maybe at least it is with people. And attractors may enable association with being in the old or the current trial. Okay. People have to remember not only that I've seen this glass or this sign with a car or this engine, but I associate it with this trial or a preceding trial. Therefore, I know whether to not answer to this one. Okay. I think Daniel Amit would have liked to work on this, and I'm sorry he's not here to work with me. Let me just add that we are doing some work together with Danny's son, Yali, and we have a couple of papers with him. Some of that is work that I've quoted here today, not only Volodya, and that's thankful. If you're interested in working on this new model or other aspects of psychophysics, come and see me. Thank you very much. Yeah.
So I was wondering if the people were given verbal instructions before the experiment.
Yes, people are given verbal instructions. The monkeys, of course, are not given verbal instructions. They are behaviorally shaped to respond properly. But the people are, they're told, wait for a match and then respond.
Do you think that might have something to do with the results?
Yes, but I'm not satisfied with that. In other words, to say that there's some homunculus there in the brain which heard the instructions and therefore set it up and set the program that way, I believe our brains are programmable, and that when we go from one situation to another, we switch programs and so on. But I'm not so sure that we set up that program just by getting verbal instructions. Or if we are, how does it work? That's my question. Even if we do it, how? What is the mechanism? What network is set up? How do we remember? You guys just saw those pictures. There were 14 trials, maybe 5 each, 70 pictures. And you remembered which one was there and which one not. How? It's amazing. Okay? Yeah.
About the pictures shown to people in the experiment, were they abstract pictures or pictures like we saw here?
In our experiment, they're pictures like you saw here. Mainly, well, there are 2 possibilities. We could have done a fractal pattern and so on, which we've done, and others, as you saw, have done with those with the monkeys and so on. So they're abstract. To the monkey, of course, it doesn't make a difference, because a choo-choo train is no more less abstract than a fractal pattern. But Yes. So it is, for the people, it would make a difference. I was concerned that when we move from monkeys to people, people would start giving names to the pictures. Okay? But it doesn't help, because they would give a name, champagne glasses, but then they saw another picture of champagne glasses and they know that it's different. Okay? Or they would say, well, there's green in there, but there's green in the next one too. So I'm not sure that the naming works, and we're showing them pretty quickly so they don't have time to, okay? One thing that people do in experiments, we want to avoid people from naming things, is that people should recite a poem or something like that, or say a word or repeatedly while they're doing the experiment. And once you're doing something like that verbally, then it's very difficult to start giving names to pictures and remember them. Okay? Yeah, fine.
Two questions. So in the monkey case, when novels after picture, so they release that mechanism, did you try to ask him for false positive? Does he remember?
Yeah, we did not. We didn't do that experiment.
Okay. And the second question, in the case of novels that the monkey is performing very well, is there any physiology?
No, there is not. There is not. All I can say, though, is that Miyashita, when he did these 100 stimuli and looked for the towers, he did another 100 which were new, were novel. And there was no delay activity. So that's where it comes from, this notion that delay activity only comes later, only comes with a lot of stimuli.
But if there is an attractor...
But exactly, once we think of attractors not being delay activity, then sure it can...
Yeah, but if there is an attractor for one presentation...
Right, that's what I'm saying. Then it could be completely different, okay? It could be an attractor which is not seen in the same neuron responding. And who says that he looked well enough? Okay, he did that. Okay.
I suppose this is another experiment you didn't do, but did you try to blur the pictures visibly, just to have the capacity of perception, and see how fast it performs the theory?
We of course haven't done it,
see the
but
details or just some of the colors?
you know, I would expect that with a little bit of blurring, people will look more carefully and therefore might have better memory. You would have to do enough blurring so that it's difficult to differentiate. Yeah. Okay. So it's tricky blurring. Yes.
Have you done anything a little bit like that to confuse, to make it a little more difficult? For example, if two less similar or less more normal or more similar.
Okay. As I say, we have done it, and the pictures you saw, they are often confusable, like the champagne glasses or the signs and so on. The one who's done this systematically is Aude Oliva at MIT, and she really she put an oven, an open oven, either with or without a bread inside, or things like that. Okay, and so she was very careful and to make sure she had 5 different cars in the series, and okay, and okay,
What is novel?
Yeah,
Context dependent.
that's right. Thank you.